Welcome, everybody. My name is Marina Heinonen, and I'm a food chemist and also a professor in food safety at the University of Helsinki. I am also a longtime member of EFSA NDA panel and the working group on novel foods.
Welcome, everybody. My name is Inge Mangelsdorf. I'm a toxicologist working in the field of risk assessment. I'm self-employed now. But at former times, I was the head of the Department on Chemical Risk Assessment at the Fraunhofer Institute for Toxicology and Experimental Medicine in Hanover in Germany. And I'm a member of the NDA panel and of the working group on novel food. Okay.
Thank you, Inge. So you are now watching a webinar on the scientific aspects to consider when preparing a novel food application. And before we start on the actual scientific content together with Inge, I would like to draw your attention to some of the technical aspects. Please be reminded that the webinar is being recorded, and also that EFSA has a policy on webinars, and should you not agree with the policy, please disconnect now. You should also note that the webinar is in English, so we would like all of the questions that you may have to be also in the English language. Just a quick check on the audio. It should be automatically connected, but please do check. Control your volume. Make sure that you enable the sound on your computer and also that you turn on your headphones. This is what you should be seeing. You see the presentation window, the presentators, and then you see a chat box on the bottom half of the view. And here what you can submit is the technical issues if the sound is not good for you, and the EFSA staff will help you on this. And then you also see a files box, and you can download available files from here, for example, the guidance documents relevant to the novel food applications. And then you also see the question and answer box. I would like to remind you here that we are receiving the scientific questions on the subject. Please try to avoid the regulatory issues. And as me and Inge, we will keep talking on the presentation, but we have expert EFSA staff to respond to your questions. When you are sending the questions to us, you can continue doing so until 1:30, and then the EFSA staff will be answering until 2 o'clock. And should that be the case that your question is not answered, please feel free to resubmit your questions via the EFSA app desk. So now about the presentation that we are about to do. So my part is first to give the key aspects regarding the compositional data and specifications on the novel food application, and then Inge will give an explanation on our toxicological testing strategy. So this is how what are the different parts in the novel food application, and this is also from our renewed guidance on the novel food application preparation. And I would like to first say that please remember that the identity of the novel food, the compositional data, the specifications, and also the production process, it's really a prerequisite for the toxicological assay, the risk assessment of the novel food. And while we are talking about the novel foods in general here, of course, this identity, production process, composition, all the demands, they also apply to the traditional foods. If we start with the identity of the novel food, of course, the data that we require depends on the nature of the novel food, whether it is a question of a chemical substance or a polymer, or if it has been isolated from microorganisms or from plants or from animals or cell cultures. And just to be also helpful in case that you are dealing with a novel food that consists of engineered nanomaterials, there's also guidance being updated currently. A few examples on how the demands can be different depending on the nature of the novel food. If we are dealing with a chemical substance, of course, what we request is the UPAC nomenclature, the CAS number, and possible other identification numbers. We'd like to see the synonyms, the trade names, the abbreviations to be able to identify the compound. And then, of course, we need the molecular and structural formulae, the stereochemistry, if applicable, and of course, the molecular mass. And from the past experience, some of the examples are with the lipids, with carotenoids, with phenolic compounds, for example. If the application concerns a novel food that is from or isolated from plants or animal material, then we'd like to see the scientific name, either the botanical or the zoological family, and we'd also like to see the information down to the subspecies with the different animal or plant species. We'd also like to see the synonyms as well as the common names of the plants and animals, and especially for the plants, we are interested whether the plant is included in an internationally recognized database. Regarding the parts used, of course, it will be different whether we are dealing with a whole plant compared to, for example, a root or a seed or a leaf or a berry or flower, or don't you think, Inge?
Yeah. So,
would you, for example, if you have toxicological studies that are on the whole plant, would you think that they are representative of a different part of the plant?
no, not at all. So we have really to assure that the relevant ingredients are being tested.
Yeah, I fully agree. And we've had cases where we have considered this.
Yeah.
And why would we need the data on the geographical origin? Maybe in the cases when there could be some environmental contaminants, pollution, and of course, if it is a question of a plant material, of course, the climate conditions will influence the composition of the plant. Regarding the production process, we are also looking for detailed information. And of course, here it depends on what we are dealing with, whether the novel food is from a chemical synthesis, whether it's a fermentation product, or if it's an isolation product from a natural source. And in this case, we also need very detailed information on the potential byproducts, the impurities, the contaminants that could raise safety concerns.
Yes, and that is also a very important point for us as a toxicologist, because we should know in beforehand which ingredients may be relevant, and what we usually at least should know are the contents of different metals, because this may raise safety concerns, but there may be also other impurities which are relevant in the end.
That is right. And of course, if we are dealing with a novel process, we need the description, the full description on the process. And the past experiences, we have dealt with UV treatment, for example, and of course the different kind of synthetical products. We also need data on the raw materials, the starting substances. Of course, in the application, you will also describe the history of the source, but also in this part, you need to describe the raw material, the starting substances. And depending on the origin, we also need information on the handling of the sources. In case of animals, we are interested in the breeding, farming, hunting conditions. With the algae and microorganisms, it would be about the culture conditions. And regarding the plants, it would be the cultivation practice, time of harvest, for example. And there's also a guidance document to help you here. Regarding the production process, also the operational limits, the key parameters are of importance. And here, if we are dealing with chemical synthesis, it would be the reaction sequence, the side reactions, the purification steps, and the reaction conditions. And if we are dealing with the plant or animal or microbial products, then we'd like to have the details on the conversion of the raw material into the novel food. What is also needed is the assurance that the production is well controlled. And what is also needed is the flow chart. And why this information? It should allow the conclusions on the impact on the process, on the safety and the nutritional value of the novel food. And of course, we treat all this information with confidentiality. Yeah. We need it, but what will appear in the opinion in the end will only be the non-confidential part. I'm sure that you are well aware of the clock stop procedure, and that means that we are stopping the clock while we need to get some more information from the applicant. And regarding the production process, we've many times gone back to the applicant when there are steps missing in the production process. We don't fully understand how you go from one step to the next one, or there's insufficient information on the source, on the raw materials, on the reagents, the equipment, enzymes, all different steps in the process. Or if no flow chart is provided, or if no production yield is provided. And we also like to see the information on the quality assurance system. Sometimes we've gone back to the applicant in cases where the data has been insufficient on the effect on the production process that has been applied. For example, I think we've had cases where there has been a novel process heating or novel process using the UV treatment, and some products have been arising, like lipid oxidation products or some Maillard reaction products, and there might be a concern on the amount and also how much these products are actually forming during the process. Regarding the compositional data, we have the general requirements. Of course, we need to know the identities and quantities of the impurities, the byproducts, the residues, the chemical and the microbiological contaminants. And then what we also need you to consider is the toxic, addictive, psychotropic, allergenic, or other substances of possible concern.
And that needs much knowledge in advance about the substance which is applied for. So this is a challenge, this step in
the
application.
You're correct. I think this is a challenge, but in order to assist the applicants, I think we have the botanical compendium where you can identify some compounds of concern. And also, if you think that there might be some other concerns that are not found in the botanical compendium, of course, the literature survey is always advisable.
Yes, we strongly recommend to provide at least a short literature survey on relevant ingredients and frequently occurring in major amounts occurring ingredients to be sure that everything is covered.
True. And then, of course, we are interested in the type and the spectrum of target analytes depending on the sources. So if we are dealing with the chemical synthesis, it would be again on the residual starting materials and byproducts. If we are dealing with the fermentation, it might be the undesirable metabolites. And if it's a question of a plant extract, for example, it might be about the residual solvents. But here, my advice would be that please do consider the source of the novel food. And some more examples on the compositional data requirements. So if we are dealing with single substances and simple mixtures, what we need to see is the identity tests, the physicochemical properties. We are also interested in the solubility data in water and other common solvents, aren't we?
Yes, and these impact also on the performance of toxicological studies because, for example, in genotoxicity testings, the substance may precipitate and also in repeated dose studies, we have to consider the solvent for the gavage studies. So that's an important point also for our part of the application.
So you see that all this characterization and
Yes,
the toxicological information, they go hand in hand. So what we also need to know about the single substances is the particle size, shape, and distribution, and the minimum purity value, and some density or viscosity parameters, especially if we are dealing with carbohydrates, for example. And with the simple mixtures where they can be fully chemically characterized, we would like to see the identities and the relative ratios of all of the components, and also the complete mass balance is requested. When we come to the complex mixtures and whole foods, and here the examples of complex mixtures could be different plant extracts. With whole foods, we have experience, for example, with the UV-treated milk and bread. And here, of course, not all of the constituents can be fully chemically characterized, but some information is mandatory. So we need the qualitative and quantitative characterization of the main constituents, at least via the sum parameters. And if it is a case of a whole food, we are looking for approximate analysis, meaning that it would be the water, the carbohydrates, the lipids, the proteins, and the carbohydrates could be by calculation. We are also looking for the mass balance in this case. And we are also interested in the micronutrients that may have nutritional value. One example could be a preparation out of algae that is high in iodine. So we would like to see that also all the way to the specifications. And then we need the information on the components that are characterizing the nature of the novel food. And here, of course, this is the challenging part, in addition to the impurities and the possible compounds of concern. Because here, for example, if I take the example of phenolic compounds, we would not be happy just by seeing a unspecific total phenolic value, but we like to see as much as possible the compounds being identified, and here use of liquid chromatography would be recommended. And of course, the number or amount of the unidentified components should be kept as low as possible.
Yes, and the difficulty in characterizing whole foods leads also us to the general demand to test the whole food as it is. There may be exceptions, I will tell later on, but that's the reason why we in toxicological studies really want to test everything and not single constituents.
And that is why also in the toxicological testing, the item that is being tested needs to be representative.
Yeah, that's also very important.
Yeah, that's important. So I already mentioned this compendium on botanicals, and I think it's very useful because it's a database on botanicals reported to contain naturally occurring substances of possible concern. So applicants can use this for hazard identification. But of course, if you think that there's data on the whole plant and your application concerns only part of the plant, this might not be relevant. But also one advice is to use this read-across principle that if you're dealing with a plant that belongs, for example, into the Brassica family, and you already know that there are other plant species having some compounds of possible concern like erucic acid or glucosinolates, it might be worthwhile also to analyze the amount of these components in your novel food. Of course, this compendium has its limitations because it doesn't mean that if something is absent from the compendium, that it's devoid of any hazardous compounds. And we also know that this compendium, it doesn't include algae or the cyanobacteria or fungi. Regarding the compositional data, there's a question about batches, and we would prefer having 5 representative batches. with the use of validated analytical methods and also with having description on the methods. We are interested in the limits of detection and quantification, and of course the references should be provided. In case of using the in-house methods, the full description and validation of the procedure should be submitted. Sometimes this can be done via the publication. So in thinking about whether you should supply data on 5 batches or data on 3 batches, you should consider the nature of the novel food, whether it's a complex mixture or whether it's a single chemical substance. And also it matters whether the proportion of the unidentified compounds is high or if the purity is high. And then it also matters whether we have a novel source that has no history of human use, or whether it's something that we have been eating, at least as a source. And then we come also to the question about the margin of exposure. And I know that Inge will come back to this issue of margin of exposure, what is small and what is high.
Yeah. So already now we can say a small margin of exposure would be below 200 or even below 100. And a high margin of exposure would be above 200. So that's at least a rule of thumb which you can use here.
Also, the stability is of importance, and usually the stability testing is done on the susceptible compounds that might change during the storage, like the carotenoids or phenolics, or it could be something, a reaction product, a lipid oxidation product that is forming during the storage. And what we'd like to see is the stability testing done both on the novel food itself, but in cases where the novel food is intended to be added to other foods, it should also be tested in the matrix. A little bit more about the frequently missing information. So we are going back to the applicants, I think, every time, at least once or twice, sometimes 3 times. And one of the reasons is that we have inadequate information on the methods and the laboratories and the limits of detection and quantification, or there's simply incomplete information on the composition. I think we fairly often go back to the applicant to ask for information on the protein content and on the methods, how to measure the protein content, as well as the limits of quantification and detection. We also go back to the applicant when we need more information on the compounds that characterize the novel food, like polysaccharides or phenolics or carotenoids. And of course, regarding the impurities, to verify or get some information of the nature of the impurities. Regarding the batch testing and the stability testing, in most cases the problem is the missing or insufficient information. And when we come to the specifications of the novel food, there might be the case where the critical components from the source or from the production are missing. Yeah. For example, if a catalyst like palladium has been used, or if we don't have the information on the residual solvents when we know that some solvents have been used, those are the cases. So I end my talk with this slide on the specifications, and I'm sure you know that this needs to be put into a table format, and it should include all of the key parameters that characterize and substantiate the identity of the novel food. And of course, there should be also the rationale why you have selected these parameters. It needs to include the minimal purity of the novel food. It needs to include the limits for impurities and degradation products, especially if there's a possible concern. We also need to have the maximum levels of contaminants, especially if there are no legal requirements. And of course, the methods for analysis is a must. And you should also pay attention that these specifications will be considered and used by the EC in the marketing authorization. So I think this concludes my part on the characterization and specification.
Mm-hmm. And you want to give us some take-home messages from your talk?
Definitely my take-home message would be that please consider carefully all the data on the composition and the production process, because it's really a prerequisite for the risk assessment.
Yes. I can really assure that we have many questions always on these issues. Okay, thank you, Marina.
Can I hand over to you?
So then I move on with the toxicological testing strategy. And here we have again the novel food guidance, and my talk deals with points 2.8 on absorption, distribution, metabolism, and excretion, and also the toxicological information. And there I also want to focus on several parts. So also I will talk a little bit about the test substance to be tested, and also some general considerations you have to do in advance. And then I come to the tiered approach in toxicity testing, which involves several endpoints, and I also will present some specific cases to you. Okay, the approach in toxicological testing, it depends on the type of the test material. You heard already before, we have different types of test materials which are either single substances, or we have simple well-defined mixtures, so these are rather easy to handle. But we also have complex mixtures or even whole foods. So all has to be covered in our strategy. And as also mentioned before, the study has to be done principally with the material which is intended to be marketed. So the test material should be manufactured according to the procedures described in the section of the production process, which we heard before. And it should meet the compositional characteristics and also the specifications. If this is not the case, and that's quite frequent then, when the applicant uses publications in the open literature for their case for supplying toxicological studies. So in these publications, the substances may stem from other producers, and also in the publications, the data may not be so comprehensive on the composition of the test substance. So there it is very important for us that you, the applicant, provide a rationale why this material which has been tested is representative for the novel food and is appropriate for the toxicity studies. So Marina, maybe you can add something on this, what we really want to have from the applicants.
Yeah, I think it's very important to follow the guidelines, to provide all the data on the composition and the possible impurities, and also to ensure that the material that is being used in the toxicological testing, it's representative.
Yeah, that's also often an issue of questions to the applicant. So, but everything has an exception, and in the case of complex mixtures, for some endpoints, especially for ADME studies or for genotoxicity studies, it may be necessary also to investigate the toxicologically relevant constituents. And the toxicologically relevant constituents are either, or in addition, the major components, or these are the components with biological or toxicological activity, which have to be decided on by a case-by-case basis. So that's one exception. And you can imagine for ADME studies, it's not possible to follow a mixture in this, its fate in the body, so you really have to have some representative substances. But there's also another condition where we may deviate from this, and if you test a complex substance, it may be the case that you can not test very high concentrations, and these high concentrations are in the end needed for having a sufficient margin of exposure. And in this case, it may be necessary either to test relevant constituents or to enrich the relevant constituents in a mixture in order to have as many relevant components at once in the study. So these are things which you have to consider well in advance before doing your studies. So then, as already mentioned before, also for the toxicity section, we really have to know enough about the chemical properties, and you have to consider these when designing your studies. And then we have also to consider the human consumption of the novel food and its source, because we have this matter of the margin of exposure, and as said before, the margin of exposure usually has to be 200. So if we already know how much humans will take up of this substance, this will help us to identify the dose levels to be tested in the toxicological studies. And this goes together with the intended uses and the use levels which you are intending to have. Sometimes maybe we have already human studies, but nevertheless toxicological studies should be done, and these will also provide information on relevant endpoints or on dose levels to be tested. Marina has already mentioned the issue of read across. That's also important for us because related substances or related botanicals or whatsoever may give us some indications on expected effects and on studies to be performed. For example, if we have reproductive toxicity for related substances, then we want to do studies on these endpoints. Okay, now I really come to the toxicological testing, and that follows a tiered approach that assures that the most important studies are done. And as you know, toxicological studies are very expensive, so you really have to focus on the most important ones, which we support. But there are cases where additional studies may be needed, and this is triggered in this tiered approach. If you deviate from our proposals or from the proposals of the guidance, it is always necessary that the rationale is provided. And then, which is also very important, if new tests are to be conducted, they have to follow the current international guidelines, normally OECD guidelines, and also good laboratory practice. So the next slide shows the tiered approach. So you see we have 3 tiers in our approach. So tier 1 covers absorption, genotoxicity in vitro, and an extended 90-day toxicity study. And tier 2 then expands on ADME, genotoxicity, and so on. In my talk from tier 2, I only focus on the genotoxicity in vivo and to some degree on ADME. And tier 3 is then reserved for special cases. But what is also important to know that are the triggers which are used for moving from one tier to the other. And the trigger which most often is used in our assessments is the trigger on genotoxicity. So if we have doubtful or positive genotoxicity in tier 1, we go on to tier 2. So the first part is the ADME, absorption, distribution, metabolism, and excretion. And the first tier is very straightforward. It's the absorption, which has to be investigated. And then the other tiers are going on toxicokinetic parameters after a single dose. And in tier 3, we have bioaccumulation after repeated administration. So the absorption from the gastrointestinal tract. So that's normally a very easy case, because usually in an application we receive a 90-day study. And if the 90-day study shows toxicity, then it is clear the substance has been resorbed because it has caused effects. So, and that is sufficient at tier 1 in our applications. A different case is if you are applying for a substance where you are considering that it is not absorbed, which also may be the case. So, and this has to be really proven. And then already studies which normally would come in tier 1 should be applied, in tier 2 should be applied, which are the measurement of the test substance in the plasma with the ROC, the Cmax, Tmax, and the half-life, both after oral and intravenous application. And then the excretion of the test substance and metabolites in urine and feces. And Marina, you will assure me, often the analytics of the substance and the metabolites is a very difficult issue, and it's difficult to achieve low borders of detection. So in some cases also radiolabeled test substance may then be necessary.
Yeah, I do agree. Even though if you can see something appearing in the urine or plasma, it might be a challenge to identify all the metabolites.
And to be sure that everything has been covered.
Yes.
Okay, so here is for completeness the ADME tier 2, which I already showed to you for the substance not absorbed. And then we have the tier 3, and there I want to point your interest to the second bullet points, the additional data to predict ADME in humans. So that means human ADME studies, which we receive from time to time, and these ADME studies may be very helpful, especially in those cases where we also have animal studies on ADME, because then we can compare critical metabolites in animals and in humans, and maybe this can help you later on to reduce the margin of exposure which is necessary for evaluating the substance. Okay, so the next topic which I want to cover, that is genotoxicity. And genotoxicity testing has two purposes. One is to investigate heritable damage in humans, and the other one is to identify or predict potential genotoxic carcinogens. And what is very important is that the endpoints to be covered by genotoxicity studies are mutagenicity, clustergenicity, and aneuploidy. And because at least the third bullet point, aneuploidy, has a major impact on the test to be taken, which I will explain in a minute. So as you already heard, for other cases, deviations can be argued. They are possible, but we need good scientific rationales for this. And as already mentioned before, but to stress it again, it may be necessary to focus on specific constituents in order to allow sufficient concentrations, high enough concentrations, to be tested. There is a guidance specifically on genotoxicity, which you can follow and which you are advised to follow. So, what are the tests to be performed? First of all, we have in the tier 1 the bacterial reverse mutation test, so the Ames test. It is done with 4 Salmonella strains and with an E. coli strain and with and without metabolic activation. And the second test, this is the in vitro mammalian cell micronucleus test. and this is specifically mentioned in this EFSA guidance. And this test is valuable because it also tests aneuploidy much better, or also than in the more traditional tests like sister chromatid exchange or chromosome aberration tests. So that's the reason why we are requesting, if new tests are to be performed, micronucleus tests in vitro. But there may be the situation that you have already in your files other tests, so then you are asked to provide these tests and provide rationale why these tests are properly conducted and helpful for evaluation of the substance.
Can I ask you something? Is it always so in all cases that if you have the positive in vitro result, is it overruled with the negative in vivo?
That's a very difficult point. So maybe I can explain it like this. So we in the in vitro assay, we test how critical metabolites are formed and how they act. But in the in vivo situation, we have much more possibilities to inactivate these critical metabolites. But you are completely right. We cannot say that's usually the case, but we also have to keep in mind why we think that in vivo situation it is better inactivated. For example, for phenolic compounds, which tend to be positive in vitro genotox tests, they usually are negative in vivo because they are excreted very rapidly. Okay. So, but it's a very important point. It's not evident per se. Okay. And so just to mention the in vivo tests which we are requesting, this is the in vivo micronucleus test with bone marrow. And for this test, we often miss information from the applicants that it has been assured that the test substance has reached the target tissue, the bone marrow. So either it may be provided, this information may be provided from information from the test itself on the number of the immature erythrocytes, or from other toxicological studies, from subchronic toxicity testing or from ADME testing. We get some information, is the compound reaching the bone marrow. But that's important to assure. But sometimes it also may be necessary to specifically investigate the target tissues. And there the in vivo comet assay has been proven as a useful test. For example, you can assess the reactivity in the gastrointestinal tract of substance. And a very advanced test system is the transgenic rodent assay, where you really get very useful information, but it is a huge test which has to be performed. Okay, so the next topic I want to talk to you is the subchronic toxicity. That is very important in our evaluations and should be provided in nearly all cases. It serves to identify adverse effects, and it allows us to identify the BMD, the benchmark dose, and the NOAEL, which are the basis for deriving the margin of exposure. The study should be conducted according to the OECD guideline 408. But recently, the OECD test guideline 407 on 28-day toxicity has been updated, and it contains additional parameters. And you are strongly advised also to include these additional parameters in the 90-day study, although they are not explicitly mentioned now in the guidance. And these and additional parameters include neurotoxic, immunological, reproductive organ effects, and endocrine-mediated effects. So I mentioned before, there may be a lack of systemic availability, and then, which has been proven in the ADMA strategy before, described before. But nevertheless, you have to investigate in a 90-day state at least the physiological and pathological effects in the gastrointestinal tract, which comes into contact with an insoluble compound. And then also additional markers of potentially adverse nutritional or metabolic effects be considered in the 90-day studies. And as already mentioned before, for whole foods, there may be problems in designing a suitable 90-day study, and there you have to avoid possible nutritional imbalances, and it may be necessary to enrich some of the substances or test individual substances. So here is an overview on examples where subchronic toxicity studies were needed and where they were not needed. So in general, they are needed for synthetic novel food ingredients. Then they are needed for plant extracts, and they are needed for fermentation products. So all products that are deviating for the source usually have to be investigated in a 90-day study. On the other side, we have those which are not needed. And so here we have a well-characterized source with a significant history of food use. We have comprehensive compositional data, no concerns from the production process, and much knowledge on the main components and whole foods. So for whole foods, the example is chia seeds. And I want to point you to cranberry extract, because you may wonder, cranberry extract, that's a plant extract. Why is there a 90-day study? Not necessary. So I would like to ask Marina, what do you think, why cranberry extract could be accepted here?
So in the question of the cranberry extract, we carefully considered the production process and the production yield, and also the exposure, both to the phenolics from the cranberry juice, the source, and from the cranberry extract. And we noticed that there was not such a difference in any of these.
Okay, so this is an exception. You see, we have lots of exceptions, and it always requires careful analysis, but we are also happy if we can get rid of a 90-day study. Okay, so I want to show you an overview table, how we would like the toxicological data to be presented. An example. And the advantage of such an overview table is that we have at one glance several pieces of information. The first piece of information is the dose response. If you look here on the body weight for the female animals, you see that the body weight after a certain dose level is reduced, achieving statistical significance. So that's the one point. Then at one glance, you also can compare the sexes, how they are behaving. And you see also here in body weight, the males also, they are decreasing, but not statistically significant. That's often the case in our studies. And then there's another issue which is also important. You see in the 100 milligram per kilogram body weight, there is no effects. Then at 400, some effects are getting on, and then at 800, we have quite a number of effects. So it's also important for us to know, there are several effects at one dose level, and maybe there is a pattern which we can explain by some mechanistic issues. And here I come to an issue which really causes much discussion, and Marina may prove this. if we have these tables, we have to discuss, are the effects which we have adverse or not adverse? And so here are several points, and all of these points have to be seen with a question mark. They are not a list of answers to be covered, but it's are all question marks. So we start the evaluation. Is there an effect statistically different from a concurrent control? But as you have seen for the body weight before, we have some effects which are dose-related that may be also relevant in the evaluation. What is a very important aspect is the effect size. The effect size, as a rule of thumb, if it's more than 10%, then it may be relevant, but it's different depending on the endpoint. Then the dose response is an important issue. This gives us also information if an effect may be incidental. And then we have the sexes which may be affected, but it is not an argument to say an effect is not adverse because only one sex is affected. And then we have patterns which we have to address. So for example, we have liver enzymes which are changed, and one enzyme may be not adverse, but several enzymes, then we think, okay, that's a relevant finding. Then there are also other points which we have to address here. Reversibility is put forward sometimes in the applications, but if you consider a novel food which is taken up regularly, so it's no issue. And then the historical control values I want to address, because it's also a matter of discussion several times. So it should provide it with the study report, and it should be from the same lab under the same conditions. We should provide statistical background on the historical control data. Often we get only ranges. The ranges are very wide. They are not useful. And then you have to bear in mind that it is the main purpose of the historical control data to assure the quality of the control group of the study. Okay, and then we have also to see that some of the findings which may be beneficial for some conditions in humans. So if you consider weight loss, so weight loss may be a very nice property of a novel food for obese people, but it's not, but it is an effect in toxicological study which we take serious. Okay, then we have the reference point. It has been measured already. We have the benchmark dose and the NOEL as possible reference points. The EFSA now recommends to use the benchmark dose. And there is a tool which is available in the internet which you can use and which makes the benchmark dose approach really feasible. You should try this with your next application. And then coming to the size of the margin of exposure, I said before 200. This 200 is composed of a number of 10 for animal to human, and it is composed of 2.5 for toxicodynamics and 4 for toxicokinetics in the case of the rat, which is the most frequently used laboratory species. Then we have a factor of 10 for inter-individual differences in humans, covering old people, covering children, and so on. And we have a factor of 2 for subchronic to chronic exposure. So in former applications, maybe there's also a factor of 100, but due to more recent guidance, we are switching more to 200 and need some justification really to use 100. So that maybe cause some confusion.
Yeah, I think that is very different from before.
Yeah. So, and a factor of 2 may be much in some cases. So sure. we are realizing this. Okay. So, and then I come to the higher tiers, which may be triggered from relevant findings in the toxicological studies, in the 90-day studies. So we have chronic toxicity and carcinogenicity, and we have also other studies which have to be performed in this case. Usually we hope to cover many cases with the subchronic toxicity, but studies on reproductive toxicity are performed quite frequently and are especially triggered if we find in the subchronic toxicity studies findings in the reproductive organs. So that's an important trigger. Okay, so last not least, I have some guidance on specific cases, which I just want to point out to you. So there is some guidance on insects, there is some guidance on microorganisms, and finally, also engineered nanomaterials are addressed specifically with the guidance. With this final slide, I want to thank you for your attention, and I hope what I've explained to you was helpful.
Thank you, Inge. Do you also want to give us a take-home message in plain?
Oh yes, I can do this. So you really have to consider this tiered approach and well design your studies also in the whole bunch, not single studies, but address a whole bunch and see the interrelationships. And we are very grateful if you would provide us with your justifications for deviations very well in advance, because then we don't have to send you letters for with questions for clarification. So that's also for toxicological issues very important.
Thank you. Thank you. So for those of you who are currently preparing or planning to prepare a novel food application, we would really like to encourage you to follow the guidance documents on novel foods, but please do also consider the other guidance documents, especially those from the scientific committee. And then we think that you might find it helpful to look at the previous opinions on previous applications. And also we had this novel food seminar in March, so it might be helpful to look at the presentations also on those. And then I would like to also to remind you that you can continue to submit questions for 3 more minutes, and the EFSA staff will respond to the questions until 2 o'clock. And following this webinar, you should be sent an email and you will be asked to participate in the survey. Please do so, because that helps us to improve our webinars. And then at last, I think we thank you for your attendance and for all of your questions. And this, I think, completes the webinar today.